speaker is Dr. Gregory del Zoppo, and he will tell us about microvessel cell matrix adhesion receptor matrix interaction in permeability, barrier, and barrier integrity.
Thank you very much, Margaret, and thank you to the organizing committee for asking me to speak on this topic. And thanks to the Institute for support over the last years. Finally, thanks to, and probably first of all, thanks to our group in Southern California at Scripps for much of the input in the non-human primate work, as well as some of the cellular work, and in Seattle at the cellular level. This is sort of the knowledge circa 1977 of the vasculature, and it's, as you see, it's basically the intricacy of the neuronal interconnections. And it was appreciated some 100 years ago that the vasculature was very important in its application of to neuronal behavior. But the force that drove us to look specifically at the microvasculature came out of acute work, and specifically out of reperfusion of large artery occlusions in ischemic stroke. And what's clear is that following reperfusion, the downstream vasculature could be reconstituted, but the question was what happens to vessels under the size of 100 microns that could not be imaged by these procedures, and what happened to the cells around them. And virtually the time of this acute interventional work, there was very little known. What was appreciated, however, in the 50s and 60s and beyond was that the cerebral microvasculature had a particularly complex setup. Is a high flow, low pressure system, and an occlusion within the system, and the branching of the system, particularly in the cortex but also in the striatum, allowed flow to reverse and not produce downstream ischemia to a threshold. Furthermore, the structure of these vessels is not as simple as an endothelial cell on a matrix, but includes the brain itself, and specifically the astrocyte end feet, as Mike Nadergaard has pointed out. So the vasculature, the microvasculature, it contains two cell types bounded by an intermediate matrix. The model system that was used for our initial experiments was intended to look at acute problems, that is, acute events that took place following occlusion of the middle cerebral artery in the awake animal. And then specifically because of the vascular arrangement, the neuroanatomy arrangement, as well as the hemostatic arrangement in these animals, they could be used to model a human disease, at least to a limit. This is in the Papio species. I would say one thing about other species, and that is that they have other advantages that this species does not. So we spent a considerable amount of time looking at the intravascular events that take place, and some of those have been described today. They include leukocyte adhesion, events that lead up to occlusion, thrombosis, and hemorrhage. And what was interesting to find was that these events would be initiated immediately upon cessation of flow in the awake animal, suggesting that while there was neuron injury, vascular dynamism was also at play, and this was not appreciated. So for the purpose of this presentation, I'm going to focus on the events within the cell wall itself, and these are virtually silent, inasmuch as the microvasculature in most organs does not announce itself until fairly late when the bulk of microvascular disease has play clinically. These events I'm going to speak about are basically below the level of the radar that most of us have. And they became very interesting because the timing of these events matched pretty much that of neuron injury within the field. So this is a diagram demonstrating some very simple experiments early on. The territory of interest was the lenticulostriatal branches of the middle cerebral artery, so the proximal middle cerebral artery occlusion producing injury in the striatum. It was clear that one could use DUTP incorporation techniques to define cellular injury as a core, and then around the core one could sample and see what events could take place there. And what's important to know is that within 1 to 2 hours of the onset of ischemia, the vast majority of cells that were DUTP incorporating were neurons. And vascular cells did not present until much, much later, when about 2% of the cells at 24 hours were endothelial cells. So endothelial cells are really resistant in vivo to the kinds of injury that we would hope to see clinically. The second point to be made here is that this is an awake primate model. This is an anesthetized rat. So the timing and the duration, the unfolding of events that take place is anesthesia dependent and probably species dependent as well. So is a sort of a tangential to our own exploration of other things, but we began to wonder whether or not adhesion of these cells, how the endothelium is tied to the basal lamina, may be important in how the cells respond, and particularly how the blood-brain barrier evolves its injury. We knew well that tight junctions were an important element in this, but the question is, can you have a tight junction if you don't have a cell that's adherent to its base? And so of consideration were the 3 beta-1 integrins, alpha-3, alpha-1, and alpha-6. And this is just an example of what happens with alpha-1 beta-1 during ischemia. Within the first 1 to 2 hours of the onset of ischemia, there's a significant loss of beta-1 and alpha-1 integrin expression in the core, and that's seen in this region here. And there's a somewhat more modest but still significant reduction in the area where there is no real clear evidence of neuron injury. That is to say, there must be some putative injury, but in the peripheral zone there is still some degree of beta-1 loss. In fact, if one looks at all of the beta-1 integrins, except for alpha-5, there's a significant loss in this area. And in the same vessels, or in vessels nearby, there's an increase in integrin alpha-V beta-3, a pro-angiogenic signal, which is important, as we can point out later. The relevance of these structures is that they tie the vessel together, but they're also signaling molecules. As you know, integrins are involved in either outside-in signaling the environment to the cell, or inside-out signaling how the cell will respond to other cues. So these are both structural and potentially signaling molecules, and they're pretty much manhandled by the ischemic event. So for all intents and purposes, we can look at beta-1 integrins as a family, realizing that they're not really identical from one alpha to the other. Second, another point to be made is that in the ischemic territory, that is in the core, as marked in these 3 animals, one sees a loss of beta-1 in the center of the core, but around the periphery, there's an increase in beta-1 transcription. Now, the alpha partner to this is not known, but a good candidate would be alpha-5, which is important for angiogenesis. Well, what about the astrocyte side of this? You can't think of the endothelium in the vasculature of the brain without thinking about the astrocyte. And we should say, parenthetically, the neuron which is beyond that, to which the astrocyte interacts or speaks. So this is the vascular part of the neurovascular unit, of which the neuron, which you don't see here, is also a player. So in order for the vasculature to work properly, it would seem that the infeet would also need to have a strong and firm interaction with the basal lamina. These adhesion receptors involved here are integrin alpha-6 beta-4 and alpha-beta dystroglycan. Now, in GFAP positive preparations, looking at alpha-6 beta-4, one could see a number of vessels where alpha-6 beta-4 was co-expressed on the end feet, but a number of other vessels where there was no alpha-6 beta-4. And this turns out to be territory specific in the brain. For instance, this is a good example of the cortex in which all microvessels of every size, from capillaries all the way up to large vessels, express alpha-6 beta-4 on the astrocyte end feet. But only a small proportion of these express integrin alpha-6 beta-4. So this is alpha-beta dystroglycan, rather, alpha-6 beta-4. In the white matter, however, this is a one-to-one correlation. So every microvessel has both alpha-6 beta-4 and alpha-beta dystroglycan. What these 2 molecules are doing is not yet worked out, but there is some idea about alpha-beta dystroglycan. During focal ischemia in the primate, within 1 to 2 hours, there's about a 40 to 60% loss in expression of alpha-beta dystroglycan on the astrocyte end foot. Now, it's important also to note that the astrocyte is the epithelial cell of the brain. It adheres to the basal lamina much in the same way that the skin adheres to the basement membrane. But indeed, there is a rather rapid and remarkable loss of this expression. This can be mimicked in vitro, using primary mouse astrocytes. This is C56 Black 6. They can be grown on wells or on inserts. And here's an example of an oxygen glucose deprivation experiment in which there's a loss of alpha-beta dystroglycan, in this case beta dystroglycan, which is not affected by inhibitors of proteases, including a protein and the cathepsin L inhibitors, but is affected, in fact preserved by inhibitors of MMPs. is GM6001 and 110 phenanthroline. And it doesn't matter whether the cells are grown on collagen 4 or laminin 1. It appears as though there is a protease-dependent loss of beta-dystroglycan, which is associated with this change. Now, why is this important? Well, it turns out that it's important because glial cells, which are banging up against the basal lamina, detach from the basal lamina during ischemia, as shown in this 6-hour preparation from the Sprague-Dawley rat. And the glial cells detach, and there's interstitial fluid which increases between the basal lamina and the astrocyte. This is an observation that's about 45 years old, but now has a potential explanation. But what about the basal lamina itself? And here I'll try to be brief. It was Gerhard Hamann in our group a number of years ago who demonstrated very clearly that within 4 to 24 hours after the onset of ischemia in the primate, there was a significant loss in collagen 4, laminin, and fibronectin content of microvessels, that is predominantly capillaries. And there was, this coincided with an increase in pro-MMP2 expression in this system. Whether or not the pro-MP2 was activated and caused these changes was a question at large. But what's important to know is that this is not the only protease that's generated. In fact, while pro-MP2 expression was also correlated strongly with neuron injury within the territory at risk, other proteases also generated, in this case by endothelium, urokinase, were not so associated. It's possible to make a number of biochemical pathway guesses at how this works out, but suffice to say, how these particular MMPs and how members of the other families of MMPs, serine proteases, cysteine proteases, and heparinase are upregulated at the same time, interact with each other, is still unknown and unclear. What's important is that, among other things, is that the microvascular expression of these matrix proteases occurs at the same time as neurons are found to express them as well. An important caveat, again, is that as Eng Lo and I showed some years ago, that the expression of these proteases in the baboon, in this case pro-MP2, may appear different in another species, such as the mouse, where it's MMP9 that's upregulated following focal ischemia. So beware of species of questions. So this leads to the next question of how these are expressed, and this is a hypothesis, a cartoon based on data from our group that demonstrates the co-expression of heparinase, cathepsin L, which I'll allude to a little bit later, pro-MMP2 and urokinase and its receptor on both neurons and on the vasculature, suggesting that for some reason yet unknown, these two structures, the neuron and the astrocyte endothelial portion of the microglia, portion of the microvessel, rather express these compounds. So directions of inquiry that resulted from this were to determine the relation of beta-1 integrin expression to permeability status of the endothelium, and whether or not alpha-beta dystroglycan also plays a role. So I'll try to speak to this swiftly. The endothelium, as you've already heard, is bound together by tight junction proteins in the CNS, and our concern was whether or not these structures, these adhesion complexes, were also important in maintaining the barrier, and whether or not they served for the barrier itself. These three components of the barrier we would now propose are the interendothelial tight junctions, the basal lamina, and the third component would be the adhesion receptor matrix interactions. To look at the beta-1 integrin matrix interaction, we chose an in vitro system using primary murine endothelial cells and astrocytes. These were grown on inserts. I should point out that these cells are plastic sensitive. It depends upon the manufacturer of the plastic and on the matrix protein you grow them on. We used both video imaging techniques, permeability studies, immunoblot studies, as well as flow cytometry studies. So a very simple example is using an inhibitor of a beta-1 integrin. We could show a significant decrease in Claudin-5 expression, the tight junction protein. When beta-1 was blocked in confluent cells, this produced a decrease in the expression of Claudin-5, but also an increase in permeability of the system to both 40 kDa and 150 kDa molecules. This was also true of other tight junction proteins, including Claudin-5, occludin, and ZO1. And if injected directly into the brain, this produced a significant increase in permeability. So blocking beta-1 integrin increased permeability across the barrier. An important new piece of information is that in vitro, the loss of beta-1 integrin by oxygen-glucose deprivation or ischemia requires the presence of astrocytes and also serum. So this reduction is most significant when OGD and serum are combined. I think these are mislabeled. And the important point of this is that when plasma percolates across the blood-brain barrier into the neuropill, it's essentially producing serum. So these are serum-free conditions and with serum. So blockade of beta-1 integrin produces a decrease in endothelial tight junction protein and an increase in permeability. Ischemia or OGD produces significant loss in beta-1 integrin. And then finally, I wanted to make a mention about astrocytes. Brian Hawkins in our group looked at the question of alpha-beta dystroglycan blockade. And here again, an alteration in water transit occurred. Okay, just to summarize, just say that OGD decreases the rate of water uptake in astrocytes in vitro. There's a direct blockade of alpha-dystroglycan on anterior cells, mimics the effect of OGD on water uptake. This is ERK phosphorylation dependent. And finally, inhibiting the proteolysis of dystroglycan or ERK activation preserves water uptake in these cells. Signaling, I'm not going to speak to, but this is the next step. And then finally, with percolation of plasma molecules into the neuropill, there's an activation of other cells, which I haven't talked about, including microglia, which can make MMP9 and also cathepsin-L. So lessons learned, there are many, and these can be summarized very quickly to say that microvessel responses and endothelial responses, as well as astrocyte responses to ischemia, are everywhere as rapid as neurons. Finally, that the astrocyte function is matrix adhesion dependent. Endothelial cell astrocyte modulation is clearly operating, but we don't know how. Implications for signaling events and clinical events is quite clear, but also there may be potential treatment possibilities. Gaps to be filled are numerous, but I'll just focus on three. One is these are acute interventions, but what's happening in the chronic situation of, let's say, lupus neurovascular disease or amyloid deposition? How are these receptors and how is their interaction of the cells with the matrix altered more or less permanently or progressively? The second is communication within the vascular segments and signaling across the barrier, that is the basal lamina barrier. And finally, what role does matrix protease generation play in all of this? Okay, thank you very much.
Thank you. We'll take some questions. You owe me 2 drinks.
So I'm Elizabeth Hillman from Columbia University. The talks today, it's been really amazing to see everybody's picture of a cross-section of a blood vessel in the brain, because we've seen so many different layers from, the endothelium. Some people have a parasite there, some people have smooth muscle. We've seen the lymphatic system, we've seen the astrocytic end feet, we've seen all sorts of different things. And this has puzzled me for many, many years, because in the brain, you go from peel arteries, peel vessels, which don't have the glial end feet, to the diving arteries, to the capillaries. And here we are all trying to figure out what's going on from the inside out, from the outside in, how vessels change their diameter, what's the blood-brain barrier. And we don't seem to have any sort of common language or common agreement on what this thing looks like.
Well, I think it's a good point. I mean, some people are smarter than others. That's number one. And other people are just more resistant, and some people have their own kind of thing they look at. I made the schematic without the parasite and without the vessel wall histiocyte, without mast cells, without microglia, which I mentioned at the last, and oligodendroglia, which clearly play roles in how all of this is generated. The purpose for our work was to look at the acute situation. So here, these cells do play roles, the other cells I mentioned. But yeah, you're right. You're absolutely right. I think to schematize it, endothelial, basal lamina, astrocyte, this part of the neurovascular unit is still not worked out in terms of how the communication should take place, and it does, and it's pretty rapid. But your point is well taken.
Next question.
Tanya Maidas from Boston. I was just wondering, because Elizabeth Dejana's group showed many years ago, like about 20 years ago, that these integrins, the beta-1 integrins, may actually be at not just cell matrix, but at cell-cell contacts.
Yeah.
And I was wondering if you've looked at that in your vessels.
Yeah, we did. This is an interesting observation, because I'm not so sure it doesn't depend on the organ system to some degree and the preparation. We cannot exclude the possibility that there's a low level of beta-1 interaction between cells, endothelial cells, in vivo. What happens in vitro is very much dependent upon the setting. Jordan Pober showed very nicely a little bit of hydrogen peroxide removes the polarization of beta-1 integrin from the base. And I'd be willing to take the criticism that we know nothing about alpha-beta district glycan polarity on the astrocyte. I mean, these cells, it could be everywhere. So I think it's a point well worth looking into.
Last question over there.
Howard Crystal, Brooklyn. I don't understand something. Is the basal lamina the same space that the lymphatics are in? And if so, how does all that transport happen through all these proteins?
Well, the person you should talk to is sitting in the front row here about that. But I will say this. Lydia Sorokin in Münster has shown very nicely in the last 4 or 5 years that the chemistry of the basal lamina is actually quite complex. And it depends upon where you are in this vascular tree, from the peel penetrating vessels all the way through the cortex down into the striatum. And indeed, the let's call it the abluminal side of the endothelial basal lamina and the astrocyte portion of that basal lamina have a different composition, and they fuse in the region of the capillary. And I think you've already heard today that the capillary has something to say about CSF production. So how this is all worked out, I think, is probably not entirely clear. And you're raising a very important point, and that is that the composition and structure of that part of the vasculature along the whole tree is going to be very important to investigate. Fair enough?
Thank you so much.